The majority of breast cancer is caused by mutations that are not inherited. However, in 5 to 10% of cases, breast cancer arises due to mutations that may be inherited from either parent. Of these cases, mutations in the BRCA1 gene on chromosome 17 and BRCA2 gene on chromosome May 13 account for 80%. There are tests available to detect certain mutations in BRCA1 and BRCA2. These mutations greatly increase the risk of developing breast cancer. First, a sample of blood is taken. DNA is then extracted and purified from white blood cells. If an affected family member has been tested, and the location and type of mutation is known, a test that looks only at that particular site, a single-site analysis, may be performed. For example, testing might have shown that the person carried a deletion in BRCA1, so another family member may be tested only for that possible deletion. First, portions of the DNA are amplified using polymerase chain reaction, PCR. The primers are designed to detect a deletion. If the person has the deletion, the blue primer binds to the left of the mutation, and the green primer binds to the right. If the gene does not have this deletion, the blue primer binds to the left, but the green primer is designed not to bind. When both primers bind to the gene, the PCR process produces millions of copies of the mutated DNA sequence that lies between the two primers. The DNA pieces can be viewed using gel electrophoresis. Some mutations are common in affected families from defined populations. If a person from an affected family is of Eastern European Jewish or Dutch ancestry, she might be tested using multiple site analysis. This test also uses PCR with specially designed primers to analyze several sites at once with no mutations. There are several possible test results. Positive test result shows that a mutation has been found and that the mutation interferes with protein function. Carriers have a lifetime breast cancer risk of 56% to 85% and an increased risk of ovarian cancer. A negative result indicates that the test did not find a mutation known to be harmful. It is likely that the person tested has the same risk of developing breast cancer as the general population. An indeterminate result is also possible. In this case, a mutation may have been detected in a single base pair. These mutations might make no or only minimal changes to protein structure and might not be associated with cancer. These tests are not for everybody. The people who benefit most from the test are women who belong to families with a high incidence of inherited breast cancer who want to know if they are at increased risk of developing the disease. The test does not catch all mutations that may be associated with hereditary breast cancer. Most of these mutations are unique to a family, so each affected family may have its own mutation. Also, approximately 20% of hereditary breast cancer is caused by mutations in genes other than BRCA1 and BRCA2.